Hello, dear colleagues. I'm pleased to present to you our topic. It is Lamprey of Ukraine as a prospective object for cryobiological research. Unfortunately, we had to present the first experimental result by the end of May 2022, but the Russian military aggression has changed our plans. However, we are ready to present our ideas about Ukrainian lampreys as a promising animal model for cryobiological studies. A little about lampreys and their evolutionary background. In fact, lampreys are living fossils that have conserved archaic body structures for 340 million years. Lampreys are one of the two surviving group of jawless fish, lampreys and hagfish, among huge variety of extinct jawless fauna. And modern classification place lampreys more closely related to vertebrates, making them only one representatives of the basal group in relation to modern vertebrates. As you can see on the slide to the right, you can see that body plan of modern lampreys is not seriously deviate from the extinction one. And on the slide of the left, you can see the huge diversity of jawless fauna of the Silurian and Devonian periods. So here you can see a big table about the popularity of lampreys as research object in different field of biology. As you can see, the numbers of publication on neuroscience, biochemistry, genetics, developmental biology, immunology, biophysics, computational biology is growing up to 2024, and such trend maintain. Of course, main argument to sell as that object is the exceptional position of lampreys in the evolutionary tree. So here it is. And but what if lampreys are interested not only by their position of the evolutionary tree? No. Lampreys live in cold water and reproduce in low temperatures of environmental. Lampreys belong to the monotypic class. There are only three families: northern, pouched, and southern top-eyed lampreys, and they distributed circumpolarity in preferring subarctic or temperate waters, as you see on the slide. Here it is. Different taxa of lampreys and their area. Most important stage of the lamprey life cycle take place in water below 10 degrees. And among them are storage and maturation of reproductive products, fertilization, both maintenance of sperm motility and the ability of the egg to fertilize. And it is also supposed that embryogenesis and metamorphosis of growing larvae into adult is more successful in low temperatures. In addition, unique phylogenetic position and so reproductive process are linked to lower ambition temperatures. As you can see, the sperm also. One moment, please. Oh, here it is. Spermatozoid, larva, and the grown-up lampreys that are maturating now. So on the slide, you can see our result of weekly temperature monitoring on the location of the spawn location of lampreys in the wild. As you can see, the temperature fluctuates only between 780 degrees and not higher. We can see the name of these locations. It's Studinov Creek near the Svetokirsk between Kharkiv and Donetsk region. Their spawning occurs in March, as you see. Okay, let's go. Typical model object worldwide is Petromyzon marinus, but it is not represented in our fauna, so we should find some local lampreys to work with them. Our fauna of Ukraine is represented by two species of lampreys. It's Eudontomyzon mary, or Ukrainian lamprey, and Eudontomyzon danfordi, or Carpathian lamprey. Carpathian lamprey represents parasitic lifestyle, and Ukrainian represent non-parasitic lifestyle. So, and we will talk about Ukrainian lamprey. Both of these species are included in IUCN Red List and the Red Book of Ukraine. Main reason, in our opinion, our team to develop method of using local lampreys includes availability to breeding stock. So we easily can take some individuals to experiment. So promotion of Ukrainian rare species, it's very difficult part of our conservation. Need of reintroduction in natural areas, and reproduction in artificial conditions as a source of material for fundamental search, exceptionally for cryobiology. Little about our species. It's the most widespread species in Ukraine, according to literature. However, our data says that actual habitat areas is strong. It has strong deficit, and at least their population has decreased significantly in numbers in recent time. And we suggest that it's necessary to create reproduction facilities for this species in artificial conditions for further reintroduction and restocking of the existing population. Such actions we propose together with standard conservation measures. You can see here our data of its distribution, but in fact, most of these points are now forbidden by lampreys. And here is main point of our field work, where we see unique population of lampreys with annual spawn. So in fact, it's only one actual lampreys location in Ukraine. We monitored this population for 10 years. And it's it is another deal, but as you can see, and population trend, it has no good prognosis for it, because we have very big fluctuations, and it's not very good for future of this population. So on this slide, you can see pictures from this creek that our team monitoring since 2012. And here you see some element of the spawn. You can see how they dig holes for spawning and use the photographer boot as support for wave-like body contractions. This is how they clean substrate from detritus. And here you can see this mating act. Now, here on the slide, you can see pictures of Susan Creek. It's cold groundwater, fillet, and it creates unusual temperature conditions for that region. Maximum 10 or 12 degrees Celsius in hot summer days. And these slides show pictures from our last expedition. And from this expedition, we may have some material from our cryobiological research, but it is not to be go. And this last expedition we can conduct in February 2022. And here you see first lampreys that prepare to spawning. There you can see larva or amocoid. Here you see male lamprey and is female lamprey, and our volunteer students who help us in field to carrying field study. So, in conclusion, possible cryobiological application of lamprey as model organism is cryopreservation of gametes and embryos for conservation too, and research mechanisms that enable proceed metamorphosis, research reach maturity, fertilization, embryo development, and hypometabolic mechanisms in winter at low water temperatures, 10 and below. And in end of our lecture, I want to acknowledge a team of our member team, Andrei Tupikov, who is now in the Ukrainian Armed Forces. He studied lampreys with us too, and he does everything possible to enable us to conduct field studies and obtain material for laboratory research in the future. Thank you for your attention.
Thank you so much for your report. It's really, the war has changed the plans of, I guess, everyone on this planet. And unfortunately, we cannot conduct experiments as we did before, but we are looking forward to the results of your cryopreservation of gametes embryos, because we need at least to preserve lampreys, our Ukrainian lampreys, for the future. So it's quite important work that you are now managing to do. And for now, let's just finish this first part of the session. And I hope that you enjoy all the talks as well as I did. And let's meet in 20 minutes, or actually a little bit less, for a second part of our conference for cryomedicine and biotechnology session. It was really exciting work of all participants, and I want to congratulate all the speakers and the audience as well. Thank you so much for this first part of the conference. Thank you, and let's meet at 1 p.m. for the second part of the conference.
Thank you, everyone. Thanks to everyone for supporting our AMI and Institute. See you after short break. And now you can see posters presentations.